Okay, so good morning, good evening, good afternoon, everyone from all over the world. Welcome to this talk on nanopore sequencing for viruses by Dr. Amanda Warr. I'll give a quick introduction. Um, Dr. Warr is a postdoctoral researcher at the Roslin Institute at the University of Edinburgh, and she is an expert in long-read sequencing, particularly nanopore sequencing. She runs Scotland's nanopore user group, Iron Brew, which is what you see there. Um, it's a it's a play on, I guess, a drink there in Scotland, um, and maybe of interest to the people here is that one of her current projects is sequencing and epidemiological analysis of porcine viruses in the field in the Philippines. And I'm personally grateful to Dr. Warr because the first time I saw nanopore sequencing firsthand was when she very generously allowed me to drop by when they visited Central Luzon State University last year. So our goal um, is to sequence SARS-CoV-2 here in Mindanao. So I've asked Dr. Warr to give us an introduction to nanopore sequencing of viruses. So let's all welcome Dr. Amanda Warr.
Thanks, Khalil. And thank you all for coming, even though it's in the evening there. My morning's only just getting started, so I appreciate you coming a little bit late. So yeah, we'll get started if my slides will progress. Go on, there we go. Okay, so as Khalil says, I'm a postdoc at the Roslin Institute, which is just south of Edinburgh here. This is what it looked like in the summer, but at the moment it looks more like this. I work in both wet lab and dry lab, so I do both the bioinformatics and the wet lab stuff. Occasionally I'll let people do some of it. So currently I have nanopore projects that are actually mine, focusing on microbiome sequencing primarily in cows. And I'm also sequencing African swine fever virus and porcine reproductive and respiratory syndrome virus in the Philippines with that collaboration with CLSU. I also help on a lot of projects because I've become known as the kind of local nanopore expert, largely because I started this Iron Brew group. So this is the Scottish nanopore user group. So I have people from all over Scotland come for workshops, seminars and meetings. We are on Twitter, so if you are the sort of person who likes to be on Twitter, you can follow us on there, and we try to retweet any interesting nanopore things that are going on. And as Khalil says, it's a play on Iron Brew, the drink. This joke tends to go over the heads of anyone who's not in the UK. But whereas in most of the world, Coca-Cola is kind of the drink that everyone drinks, in Scotland, Iron Brew is the main soft drink. So we went with a very Scottish pun for our user group name. So I'm going to do a quick introduction to nanopore in general. I'm kind of hoping that most of you have some idea of how it works and what it is, because we don't have a lot of time. And then I'm going to go over library prep specifically for SARS-CoV-2. I'm not going to just read off the protocol. I'm not going to go into massive details of exactly what you're doing at each stage, but I'm going to cover anything that I think you need to know that isn't in the protocol primarily. And then I'm going to introduce you to the sequencing software, which is called Minnow. And then I've got a section on practical problems, which is basically things that you don't really know that you need to know until you know them. And then I'll briefly touch on some bioinformatics. I know most of you aren't bioinformaticians, but just sort of the very, very first stages of it, it's good to know. So hopefully get everyone feeling ready to get going with sequencing SARS-CoV-2. So Nanopore have several different platforms. The one you're probably using is this one, the MinION. There's also a big brother of this one out now that's got a touch screen on it and the compute built in as well. But the MinION plugs into a computer or a laptop. The GridION is kind of 5 MinIONs set on a computer. And that's kind of the main difference between the two, is that this one's got a GPU and some compute built in and 5 slots on top for MinION flow cells. The PromethION is the biggest one, and it has either 24 or 48 flow cells, depending on which model you buy. And each flow cell on this one can produce, I think they've gone up to 180 gigs of data now, so they're really high throughput, whereas the MinION one produces maybe up to 30 gigs. That's for a really nice library. So you could probably expect somewhere between 10 and 30, depending on how nice your library is. And then we've got the Flongle flow cell, which is the mini flow cell. So this actually sits in a MinION. And this one is disposable, whereas the other flow cells, they do expect you to send them back. But the Flongle is actually disposable. And this is really designed for kind of applications in hospitals and that kind of clinical setting. It's very low throughput, it's 1 to 2 gigs. But if you're just sequencing a virus, you don't need 1 to 2 gigs. So it's still pretty applicable here if you're interested in trying that. It has got a very short shelf life is one thing to note, but it's quite a nice little flow cell. So I've got a video here from Nanopore of how Nanopore works in case anyone isn't familiar with it. It's got some jazzy music. I will try and talk over the music. Please let me know if you can't hear me. But yeah, we'll just start this video. So this is a flow cell in a MinION. And this is your library. This is your DNA with your adapters on it and your motor proteins on it. So the motor protein is like a helicase that unravels the DNA at a constant speed, which is important for the sequencer. So inside this Warr array, you've got all these wells, which have pores in them that are held in a membrane. And on the membrane, you have these little tethers that help to guide the DNA into the pores. And this is the motor protein unraveling the DNA as it goes through the pore. And as it goes through the pore, it disrupts the ceramic current. And it is the disruption of the ceramic current that actually affected with the mentioned flow band and mixes all at the same time.
Not sure why the sound of the video is too strong.
Hold on a moment, let's see. Right, it's quite low quality now, but I'll tell you what, I will I'll put the link to it in the chat later, if you remind me, and then anyone who can see that can watch it later. It's quite a nice little video, it should be fairly self-explanatory. But we'll just move on now. So general things to remember with nanopore sequencing in general, this isn't really specific to this protocol. If you are going for very long reads, which the sequencer can do, you need to be very careful extracting and handling your DNA, or your RNA in fact. You can't sequence anything longer than what you have. So if during the extraction process you break any strands, obviously you're not going to get any reads that are longer than the strands you have. You might want to consider wide bore pipette tips, or even just cutting the end of your pipette tips off with a blade, just because the smaller hole will shear your DNA more. And also do your pipetting quite slowly. It can be a bit frustrating doing it super slow, but it does help. Try to do your quantifications with a qubit or something similar. We like to call the nanodropper a random number generator, because it's quite often wrong. And it is quite important for the flow cell to have the right molarity going onto the flow cell. If you have too many molecules, you will exhaust your flow cell, and you'll end up with much lower throughput. So it's important to know what you're putting on. And also some plastics have been shown to have a negative effect on the flow cell. This was a couple of years ago at a conference. I heard that someone had used some tubes that they had lying around that weren't Eppendorf's, and it just completely killed the flow cell. And then they did an experiment where they had an Eppendorf and one of these other tubes, and it was the tube that was doing it. So this was big news a couple of years ago, but it's worth noting now. I don't know if Nanopore have addressed this since then, but if you can, try and use an Eppendorf, particularly during and after the final cleanup in the library prep. So for keeping costs down, do reuse your flow cells. You can wash your flow cell and reuse it. There's very little carryover possibly no carryover. I won't say there's definitely no carryover, but the carryover is very low. And basically the wash kit uses a nuclease to digest your previous library. So when you put a new library on, there shouldn't be anything in there. If you need to share your DNA, which is something that is recommended in some of the protocols, do not use a Covaris G-tube as it suggests in the protocol, because those are horrendously expensive. I won't buy them. You might want to share because the throughput is optimized at 8 KB. So if your reads are 8 KB, you will get the best throughput. But often you're either sequencing amplicons of a specific length that you're trying to sequence, like with this protocol, or you're trying to get the longest possible reads. So people don't frequently share their DNA. But if you do want high throughput on something mid-range, it's worth noting that you can use a hypodermic needle. You don't need to use a G-tube. And there are a lot of people who have shown different shearing sizes with different hypodermic needles online. You can find those resources. After you've run your first flow cell, keep hold of it. Is really useful for people who have never loaded a flow cell before to have one that is dead to play with and actually try the protocol on. Because obviously this is kind of the most expensive part of your whole experiment is your flow cell. And it's really, it can be a bit anxiety inducing the first time you do it. And it's really helpful to have sort of felt the whole process before you actually do it on a real flow cell. So I always keep 2 or 3 really old flow cells on my bench for new people to have a go with. You live somewhere hot. You're probably aware of this. It's hot where you live. And I'm sure you're used to protocols saying room temperature and not necessarily meaning room temperature. But definitely bear in mind that it's referring to British room temperature. And do keep your RNA and in fact most of the reagents on ice after they're thawed. One thing you might not have thought of is that the MinION does not do well in heat either. So it's very good at bringing itself up to temperature, but it cannot cool itself down. So when we were in the Philippines, we had these little mini desk fans, just USB desk fans pointed at the MinION the whole time. And this is a 3D printed stand that allows for better air circulation around the MinION. So this is actually a problem. If your MinION is too hot, if it can't maintain its temperature, the DNA actually moves faster. And then the data you're getting off it is not what the base callers are expecting to see. You end up with lower quality data if your DNA is moving too fast. So it's quite important to make sure that it stays at the right temperature. And the software does actually have a plot showing you the temperature over time that you can keep an eye on. And this is one of the flow cells that we ran while I was in the Philippines. We actually put it on quite late in the day, and this lab does not have air conditioning overnight. So this is the point where the air conditioning turned off that evening, and you can see it was a little high. It wasn't too bad, but it did struggle to maintain its temperature. So in the morning, the aircon came back on, and then poor British me arrived after a 40-minute walk in the sun to the lab, and I was absolutely roasting. So I borrowed the fan and did not notice the temperature creeping up because I had stolen the fan. So yeah, that was the moment when I was like, oh, oops, and put it back. So it does make a difference, and it is probably worth, if your lab has, if your air conditioning isn't always on, or if your lab maintains the room temperature a little bit higher than in the UK, it's worth keeping an eye on this and making adjustments. So Nanopore's protocols, as I said, I'm not going to read through everything that you do in the protocol because I think that would be quite boring. But they have very good protocols. So on the website, they have like a long version of the protocol that has a lot of little hints and tips throughout it. And I really recommend that you read the long version before you do anything. And the first time you run it, I would take the long version of the protocol into the lab with you. But once you've kind of gotten more familiar with it, they have these checklist protocols. So the checklist protocols have a list at the start of everything you need to make sure you have. And you can take these into the lab and just check off every point and make sure you've got it all. And then again, you can check off steps as you go, and it's kind of just a short form version of the protocol. And they're really useful to keep around. They've got a little spot for flow cell number and DNA sample that you're using. So you could just stick this into your lab book. I really like these, they're really useful. You might see people talking in code when they're talking about Nanopore, and it can be a bit confusing. So if you see people or hear people referring to an R number, they are referring to the type of pore they are using. So Nanopore quite frequently bring in new kinds of pore as they improve their chemistry. So you won't see many R9s around anymore. R9.4 is the most widely used right now, and R10 is the newest one and the most accurate one. The basic difference between them is that they are different shapes, which allows for more accurate, sort of more distinct disruptions to the current. So that's what those are. The ones that start with SQK are the sequencing kits and are what are on the protocols. You will probably be using 109 or 110. RAD is the rapid kit. But yeah, if you see these, that's kind of what they mean. And if you look them up on the Nanopore community website, you will get it in plain English. Oops, which one they're talking about. And then the FlowMin are the IDs for the flow cells. And this is where the confusion comes from, because the flow cell number and the R number are often used interchangeably. And you can use them interchangeably because there's a different pore in each of these flow cells. The only thing is that for this one, the FLG, that is for the flungal. So the different flow cells for the different platforms use the same pores. So that's the only time that it's not really interchangeable. But yeah, these codes can be a bit confusing. Don't worry about them too much. You're basically going to be using one of these kits, one of these pores and flow cells and kits, and these are the most accurate ones. But I think for the protocol we're talking about today, you're probably going to be using the 9.4s, the 109 kits, and the flow cells that use the R9.4s. So this is kind of the typical library prep. So you're taking high molecular weight DNA. You might want to fragment it, you might want to not. You do an end repair and then ligate on the sequencing adapters, and these sequencing adapters have the motor proteins on them, and then the tether sticks to this other end here. So the difference between that and what we're doing today is there's a little bit before it. So you're starting with RNA, you're doing a reverse transcription, you're doing a PCR amplification across the entire genome. But then from that point on, it's pretty much exactly the same as a standard library prep, just with barcodes added in. So basically, if you can do this, you'll be able to do that with anything you want to do in the future as well. So the general idea behind this approach is that we're using the reverse transcription, and then we're doing two different PCRs. So we're doing tiled amplification across the genome in two separate pools so that they're not actually overlapping within the same pool, because obviously then you would end up amplifying a bunch of this overlapping region instead of the full amplicon that you're looking for. So these are quite short amplicons of 400 base pairs with 20 base pairs overlap. And this is to increase the chance that we'll get the amplicons from most of the genome, even if the sample is quite degraded or if we've got quite a low titer sample. If you want to do this with different viruses, it's fairly straightforward to do. So this is actually what we're using for African swine fever virus and PRRS virus at the moment. If you want to design the primers, the tool is called Primal Scheme. It is available on GitHub, and there's also actually just an online version where you can just put in your sequence alignments and it'll spit out some primers for you. If you change the amplicon lengths, which is something we have done, you'll need to tweak the PCR cycle lengths to make sure that it's got time to amplify the entire amplicon. So we've been doing 7 kilobase amplicons for African swine fever virus. It's a nice stable DNA virus. And 4 kb or 7 kb amplicons for PRRS, depending on the titer. So with your input RNA, again, I'll reiterate your RNA should be kept on ice. I'm sure most of you know this. And don't forget your negative controls. You should take a negative control right through the entire process. These are the dilution factors depending on your CT values. And this is quite important. to make sure you're using the right starting concentration. So I'm assuming you guys are using samples that you've already done a qPCR on and you have some idea of the CT values. So make sure you dilute them down. This is all in the long version of the protocol, so don't worry about writing this down or anything right now. The primer sequences for Arctic Network are available on GitHub. Again, it should be quite easy to find this link. These are the same files that the bioinformaticians need. There's a few extra files on top of the primers in this GitHub, so everyone needs to know where these are. The primers should be a concentration of 100 micromolars to begin with, and then your working stock, you can dilute that down to 10. So we're using equal amounts of each primer in 2 separate pools. So just remember that all the odd numbered primer pairs go into pool A and all the even numbered primer pairs go into pool B. So again, you're diluting these down 1 in 10 to make your working stock. So the process, you're doing your reverse transcription. You can do this in a plate. And then your PCR, you want to keep your A and B separate just to make sure there's no cross-contamination there. And then once you've done your PCR, there's an ampule cleanup. I'll get back to the ampule cleanups in a minute because there's one thing you really need to remember for that. And then you've got your library ready to, your DNA ready to start your library, your amplicons. So at this point, you're expecting to have at least 50 nanograms total. You should be able to get that for most of your samples, obviously except your negative control. So these are the kind of outputs you would expect from your PCR. Again, this is in the long version of the protocol. You can double check this later. But you should have plenty. So this is at 50 nanograms per microliter here, and you only need 50 nanograms total. So for most of these samples, you should have plenty. So for the actual library prep, if you are using different amplicons or a different sample altogether, just note that you should start with 200 femtomoles here. For this protocol, you're starting with 50 nanograms. So for the end prep, the protocol will tell you to run it at each temperature for 5 minutes. I have always done it for 30. And this is because NEB recommend 30, and in the very early versions of these protocols, it did not work at 5. And Nanopore had really just cut it down to 5 so that they could boast about how fast their library preps were. I've always done it at 30 minutes at each temperature. Whether or not that's still necessary, I'm not sure, because Nanopore have made a lot of changes to their chemistry since then. But we are still doing that at 30 minutes each temperature. Then you've got your barcode ligation, another ampule cleanup, then you're pooling your samples. You can pool up to 24 samples. I don't know if you guys have the 2 barcoding kits or if you just got 1 barcoding kit, but generally for starting off with, I would go less than 24 and just see how it goes. For the first library, maybe do 10 to 12 and see what it looks like and how even the coverage is and things. But once you've pulled however many you want to pull, you've got your adapter ligation as described in the protocol. And then you've got another ampule cleanup, but it's a little bit different. You need to make sure you do not use ethanol. If you use ethanol at this point, your library is dead because you have your motor proteins in there and the ethanol will destroy them. And you also want to make sure you resuspend in the elution buffer and not water. All the other ampule cleanups, you've been using your ethanol and then resuspending in water. This one you want to make sure it's the EB, just because the osmotic balance is so important in the flow cell. The pores are quite sensitive to the chemistry, and you need to make sure you don't dilute anything down that you shouldn't. Again, you want to quantify with your qubit, aiming for 15 nanograms total. If you have more, do dilute it down. As I said, you can put too many molecules on the flow cell and you don't want to run out of fuel. So this library can actually be stored short term at 4 degrees C. When I say short term, I mean a few months, maybe 3 months. Longer storage can be done at -80 as well. So if you do end up with loads and you dilute it down, you could then just store the rest at -80 in case you want to do more sequencing later for whatever reason. It's quite handy to still have that around. So this is what your flow cells look like. And I've just labeled all the different ports here. So this is where the actual pores are. And this is the spot-on cover, which covers the bit where you actually put your sample in. There's a port just here. And then we have our priming port cover here, which swings open this way. And just under there, we have another port. I will discuss the difference between these two ports in a moment. We also have a waste reservoir here. So basically, you've got your port here, and then there are channels going up this way, across this way, through the array, up that way, and then back that way towards the waste port. So as you put in fluid here, the fluid is running around here, that way, and into the waste port. The waste port is only open when this is open. This swiveling door not only reveals a port here, it also opens the channel just here. And that's important to know later. If you are to remove anything from this side, you want to make sure it's isolated from that side. So this door being closed basically separates these two sides of the flow cell. So when you're priming your flow cell, which is kind of the first step after library prep, you want to pull out any bubbles. So bubbles are death for a flow cell. If the bubbles get into the pore array, they will kill any pores that they touch. The pores have to be constantly submerged or they will just die. So when you first get your flow cell, you see here we've got some kind of yellowy, greeny fluid in all of the channels at this end. You can see this is the port having swung the cover open, and you can see here there's no yellowy orange just here, sorry, yellowy green, don't know why I keep saying orange. There's no yellowy green just here, so that means that there's an air pocket just here, and you need to remove that before you try and put anything in through that port, so you don't push the bubble towards the pore array. So what you're going to do is you're going to take your P1000 pipette, place the tip on this port, and then turn the volume up on your pipettes. Do not try and use the plunger to pull the air bubble out. You're going to turn the volume up on the pipette to suck the bubble out very gently. And once you've done that, once you've got your greeny fluid kind of level with the top of this port, you're going to mix together some flush tether with a whole tube of flush buffer, and then put 800 microliters of it through this port here. And then you're going to leave it alone for 5 minutes. While it's sitting for 5 minutes, you can do the very last step in the library prep, which is just adding some buffer and loading beads to your library. The loading beads are extremely annoying. They do not stay in solution. They will fall out of solution in front of your eyes. Even in the pipette tip, you can watch them fall out of solution. They're extremely annoying. So when you add them, don't bother to mix them until immediately before you're going to put the library on the flow cell. Because all that's going to do is give more opportunity for you to share your DNA. You may as well just wait until you're about to put it on the flow cell. So then there's another priming step. This one can be a bit confusing the first time you do it, because the first thing you need to do is open this cover. This cover is just a bit of plastic on a sticker. It's just stuck on here. So you're just going to pick this up and flip it over and have it sit just here. But you're not going to put anything in through this port yet. You're going to put it in through this port again. So you're going to add another 200 microliters of the same stuff you added 5 minutes ago, very slowly through this port. And the idea behind that is that it raises the meniscus in this port so that the fluid is flush with the top of this port over here. And you want to do it slowly. If you do it too fast, the fluid will squirt out of that hole. So you want to do it quite slowly and just watch this port, and you'll be able to see the fluid kind of bubbling up and then siphoning back in, and then bubbling up and siphoning back in. And that's exactly what you want to see. There is kind of a correct speed to do it. You'll get the hang of it over time. Then once you've done that, once the meniscus is raised, you're then going to mix your library by pipetting up and down, just gently. And once the beads are all resuspended, you're going to take your library, hold your pipette at a 45-degree angle. This image does not show that, but it should. At a 45-degree angle over this port, and then you're not going to touch the port with your pipette. You're just going to drop the library one drop at a time onto this hole. And because that fluid is so high in that port, it should just siphon straight in. Again, there is a kind of correct speed to do this. You don't want to drop the next droplet until the first one has gone into the flow cell. If you do it at the right speed, you will see your library spreading across the entire array. If you do it too slowly, they tend to clump at this end. It's not the end of the world, it'll still sequence, it's just a little bit annoying, and it'll be more efficient if it spreads across the whole thing. So it's just kind of a drip, 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 drip. You will get the hang of the speed of that. So then you're going to close both of your ports again. This one, again, swivels closed this way. This one, you just flip it over and just make sure the little bung goes into the hole there. And then that is you ready to sequence. So the software we're using to sequence is called Minnow. It has a very nice and friendly user interface, and in fact they just updated it, so it's got a user interface I'm not entirely familiar with, so it's a little bit strange me talking you through it because I haven't had a chance to use it yet, but I'm familiar with the old versions, so it's fairly intuitive. So this can be downloaded from the Nanopore community website. It's important to make sure your computer is not set to install updates automatically, because Windows likes to shut down your computer in the middle of a run, and that's not helpful at all. You don't want to leave your sequencer running and come back an hour later to find that nothing has happened because it restarted right after you walked away. So usually you need admin rights for that. So that is something you really need to make sure is set up. So this is the Nanopore community website for anyone who hasn't seen it. You will find Minnow in software downloads here. It will be called MinION software just because Minnow is the name of the software in every single platform, and it gets a bit confusing. So on here, it'll be called MinION software. So this is what the kind of home screen looks like for the Minnow software. Again, it's fairly straightforward. You probably won't need all of the things on this screen, but again, this is on the website if you need to refer to it, but it is fairly intuitive. You should be able to sort of find your way around this screen. The first thing you need to do though is go into the hardware check, this one here. Once you've plugged your MinION in, I don't think you'll even get to this screen until your MinION is plugged in. So you're going to go to the hardware check, and this is the configuration test cell which comes with your MinION. So it's just like a white piece of plastic, and you need to put that into your flow cell and then do this hardware check, and that's going to check that your MinION is working properly and that your computer and software are set up properly. And once you've put that in and hit start, all future images of your MinION will have this little check mark next to it to say that it has been checked and it should work fine. You also have this sequencing overview. Once you put a flow cell in, you'll be able to see this. And you'll notice that this one has a little orange question mark next to it and says flow cell not checked. Again, you need to check your flow cell before you sequence it to make sure that it is up to standard. And that is using this flow cell check button here. So for a MinION, there should be at least 800 healthy pores available. If there's anything less than 800 healthy pores available, Nanopore should replace it automatically. You just need to tell them, hey, I've done this. There are only 500 pores or something, and they will just send you another one. This doesn't happen very often anymore. This used to happen quite often, but it should be fairly reliable now. And 800 pores is a really pathetic flow cell, so you'd expect to have quite a lot more than that as well. So there'll probably be more like 1500 to 1600 pores, and if you're lucky, maybe even more than that. Then when you're ready to start sequencing, so you've done your whole library prep, you've loaded your flow cell, you've plugged in your MinION, you're going to start sequencing. You need to add an experiment name. Most of this should be automatic on the MinION. On the Flongle, it is not. So on the Flongle, you need to tell it what kind of flow cell you're using and what the ID on the flow cell is. But for MinION, it should just know what you've put into the sequencer. Then you also need to add a sample ID of some sort. It says sample ID, really it's a library ID. If you're going to be barcoding things, sample ID isn't really appropriate. So just some kind of library ID here. And then you're going to continue to kit selection. And again, these are all the SQKs that I mentioned before. Which one you used will be either on the protocol you're using or on the box of the kit that you're using. So just select the one that you used. It is important to get the right one. and also the barcode expansion pack. So you probably have native barcode 104 or native barcode 114, which are the two kits that contain 12 barcodes each. So just check which one you've used and make sure you tick the right one. For this one, you probably want to turn down the run length. You probably won't need more than an hour or two. So you could set it to 2 hours so that it just stops itself. Even if you're not keeping an eye on it, it'll just stop before it goes too crazy. The bias voltage, if you have never used the flow cell before, just leave it as it is, it's fine. If you've used the flow cell quite a lot, 12 hours or more, you want to change the bias voltage. This is just the voltage that it starts on when it starts sequencing, and at minus 180 it's expecting a new flow cell. It's just how the sequencer works. And when you are running a flow cell for like 48 or 72 hours, it's going to automatically change its own voltage, but it doesn't necessarily know how long you've already used the flow cell you're putting in. So you want to make sure that if you've used it a lot before, which you probably will at some point find you've used one for more than 12 hours, just keep track of how many times you've used it, and make sure you change this. So then it will ask you if you want to do base calling. So this is a bit of a judgment call. I usually don't do base calling while it's running because Minnow doesn't tend to keep up. So the sequencing is faster than the base calling. I tend to just take the raw data, transfer it to another computer, and then do the base calling myself later. The downside to that is you can't do any real-time analysis, which is quite a nice feature of nanopore sequencing. And I'll go into why you, in particular, for this protocol, might want to use real-time base calling. And if you do use real-time base calling, you're going to want to use the barcoding as well. But yeah, you may. It's up to you whether or not you want to just run it based on time or run it based on how much coverage you have. And I'll explain how you would know how much coverage you have later. So then the next thing it's going to ask you about is the output. This is the default output location. We tend to just keep it as that. And then the raw data is the fast fives. Make sure you always have that, because no matter what, you'll be able to do base calling to a higher accuracy every time a new base caller comes out. So it's really useful to have the raw data stored somewhere. If you have base calling turned on, you could say that you want the FastQ file as well, which is the base call version. You're unlikely to have aligned it to something while it's running, but if you have, you can also get the BAM file. And you can also enter some options for filtering based on the quality or on the read length. But again, this is the sort of analysis I prefer to do later on a separate computer. So then you can save these settings so you don't have to go through this whole process again, which, other than changing the bias voltage, if you've been running it before, is probably quite a sensible thing to do. And then you just hit go and it's ready to go. So the first thing that Minnow is going to do is run a MUX scan. So there are 2048 pores in a MinION flow cell, but each sensor is only responsible for 4 of those. So you can only actually sequence with 512 pores at a time. So the MUX scan is going to pick the best pore and start sequencing with it. And then it will remux throughout the run. And this looks quite strange. You will see various colors going across the screen and you'll think something's wrong, but it's fine. Just give it 10 minutes and come back and see if it looks okay. But you do want to check the number of active pores that the MUX scan reports, because if it is different than your flow cell QC, you're going to want to try and fix that. So you can basically retry the flow cell, the MUX scan, sorry. You could restart your computer, but there may be a difference if you've had any kind of mishap loading flow cell. If you've got a bubble in there or something, there will be a difference between the QC and the MUX scan. So when you're monitoring sequencing, you'll see an overview like this. Basically, it's telling you what state each pore is in. So light green is good, dark green is okay, and then the blues basically mean they're not currently sequencing. So you want most of your pores to be in the light green. This is that same information, but this is actually representing the pore array on the actual flow cell. So for example, if you had a bubble in the top right-hand corner of your array, you would see a big blob of blue here of dead cells. So this is a physical representation of your pore array. And it tells you the same information, but you can actually expand this to have more information. This is only really going to be necessary if you're having a problem and you need to troubleshoot it. And there are various troubleshooting guides on the community website to work out why your library might not be working. But amplicons are extremely well behaved, and you'll probably find that you don't have a problem. And this is that same thing, same information again, just shown over time. So this is what state the pores were in over time. And this is quite a normal looking duty time plot. But again, if you have a problem, what this duty time plot looks like is a really good diagnostic tool for why your library hasn't worked. You'll also see other things like read length histograms. That's not particularly useful for you because you'll be seeing a nice big 400 base peak just here. But if you're sequencing something else, that can be quite interesting. And there's a lot of other plots as well, depending on what settings you put on. But I won't go through every single one of those. I'm going to speed up a bit because I'm noticing I'm running reasonably low on time. So I use RoboCopy to copy data off. I always have this stuck on the wall next to the MinIONs in case any of the wet lab people are doing sequencing, so that they know exactly how to get the data to me and tell me, hey, I've done this, go analyze it. So it's useful to have that just somewhere so everyone can see it, because I know a lot of the wet lab folk are a bit allergic to the command prompt, but it's fairly straightforward. If you need to stop a run, you just hit stop. You can either stop just the sequencing and let the base calling keep going, or you can stop everything. Right, I'm just going to go through some practical problems. So these are those things that you might not realize you need to know until you need to know them. So you might find when you plug your MinION in, nothing happens. If the fan and the lights on the MinION come on, then the cable is fine. If not, you might want to try a different cable. So most external hard drives these days use the same cable. So you probably can find the cable somewhere in the building. You may need to unplug and plug back in a few times, close and reopen Minnow, turn the computer off and on again, the usual things you might try. If these things don't work, you need to contact Nanopore because there's a problem. Bubbles in flow cells. If it's brand new and there's a bubble there, you want to send a photo to Nanopore and they'll replace it. If you put it there, try not to panic just yet. It depends where the bubble is, how much trouble you're in. So if the bubble is after the pore array and your QC is okay, don't worry about it. It's not going to cause anyone any problems. If the bubble is near the priming pore, as we discussed earlier, you're going to put your tip on it and turn the volume up. You want to keep an eye on this channel here, because as you pull fluid out at this side, it's going to pull back this way. And you don't want it to go so far that it reaches the pore array. It probably won't if you're being careful, but if you were to try and use the plunger to do this, it probably would happen. I've seen it happen with very nervous people loading the flow cell for the first time. So try not to do that. And make sure this port is closed while you're doing that, so there's no chance of you pulling air in through this hole here. If there's a bubble in this area here, the best bet is to just try and load your priming solution really, really slowly so you don't shift it. Often they will just sit and not move, but you want to keep an eye on it and be as slow and gentle as you can. If the bubble is already in the pore array, there's nothing you can do to save those pores. But what you can do is be very gentle and slow with any further moves you make to try and keep it from moving, and keep the device level so that gravity doesn't encourage it to move around either. And if you are priming and you see a bubble, do not panic. Keep your plunger where it is, remove the pipette, and then assess where it is. Because a lot of people, when they see a bubble moving, they panic and just let go of the plunger, which is the last thing you want to do, because then you're just going to pull all the fluid out of your flow cell. And it's a sad, sad thing to happen, but it does happen. So yeah, just be careful. Bubbles do appear during sequencing. They're very small and they're usually not a problem. They're usually so small that they don't actually reach the pores. They're just right on the surface near the plastic. If you try and load your library and it doesn't go into the flow cell, there's a few things you can do. This is quite rare, but it does happen. Pick up the library, put it back in the tube. If you have more flush buffer, you want to add it and try and get that meniscus up, because that's probably the problem. And then try again. You will usually have a little bit left. You'll usually have about 200 microliters left in the tube. If that still doesn't work, put your library on the port, put your tip on here and turn the volume up. But keep an eye on the library, and the second it starts moving, take this tip away to release that suction, because you don't want it to keep sucking through the port and pull air in after the library. Finally, if it really won't go in, you can pipette directly into the port. Just do it really slowly and very carefully. Don't introduce any bubbles. If there is a bubble under the port, you can actually put your library in through this port. It just might not spread as efficiently across the flow cell. This is an example of a duty plot showing you that there's something wrong. This is what it looks like if you overload the flow cell. There's too many molecules on here. I'm running out of fuel. You can see it plummeting. You don't expect to see that. So if you do see that, you can refuel your flow cell. And there are instructions on the community website on how to refuel, and you see that it pretty much entirely recovers to where we expect after you refuel. And you can see the corresponding effect of the throughput stopping, basically it's stalling there, but it'll pick up again. If it looks even more of a plummet, if it just plummets to blue here, you might have a contaminant in your library that's damaging your pores. So very quick touch of bioinformatics. There's way too much to go over now, but there are tutorials on the website. There is a tool called Epitome that will run some simple pipelines for you. And if anyone's interested during the Q&A, I can show you one of these pipelines. If you're completely allergic to bioinformatics, it's quite nice. You just tell it where the fastq file and it does everything for you. Rampart is how you would monitor your coverage in real time during the run. So this is a really nice tool that a lot of people use, and this is the only reason I would suggest you do real-time base calling for this protocol, because in real time it will align your amblicons and demultiplex your barcodes. So this plot is showing SARS-CoV-2 and the coverage over every amblicon for every sample. So you could watch this, see when you've got exactly the amount of coverage you want, stop the flow cell, wash it, and put a new library on. So you're being as efficient as possible with your flow cell. And it's also just quite fun to watch it. But again, it's up to you whether you want to be that careful with your flow cell, or if you just want to say roughly an hour is enough, and then wash and reuse your flow cell. It's up to you. So Guppy is the base caller that most people are using at the moment. This is the same base caller that's built into Minnow. You can just use Minnow on another machine to do the high accuracy base calling. But if you have high performance computing, it's much better to do it there. And then you can parallelize it. So this is the example of the command you would use. Make sure you're talking to the wet lab team so you know what flow cell and kit they're using. Guppy fast is what you would use if you're doing real-time sequencing, but then later on you want to do the high accuracy because it is much more accurate. If you don't have access to a GPU, this is really slow, but it's probably worth it for the accuracy. Bonito is even more accurate. Even with a GPU, this takes days. It's a pain, but it's really accurate. So with the latest chemistry and Bonito, you can get up to 98% accuracy, which is completely bonkers. Like, this is really nice. For demultiplexing, Guppy can do that. Guppy barcoder can do that. This is like an extra tool. If you've already done your base calling, you can use the barcoder on it afterwards. And PoreChop is a community tool, which I really like. It works very well. Arctic, the pipeline for the SARS-CoV-2 sequencing, the Arctic network pipeline, has very specific recommendations for using Guppy barcoder. And those are all in the SOP. And this is where the SOP is. Very easy to install, full instructions on the page. It only takes 2 commands and your data will be a finished genome. You may also consider using Pangolin. This will assign your new genome to an outbreak lineage. So the recommendations for you guys specifically, use your fast base calling if you want to use Rampart. If not, you're aiming for 100,000 reads per sample. You want to use high accuracy Guppy for the pipeline. You want to use the Medaka version of the pipeline. So there's 2 versions of the pipeline. The Medaka one is much faster. And remember to keep your MinION cool. That is the main message for the actual wet lab side, I think. So there's a lot of support available, very active community on the website and on Twitter. Nanopore do have a support line you can email. And there is the Nanopore community meeting just finished, but it is still available online. So do go and watch some videos on there. There's also a very detailed walkthrough of this protocol, which is why I didn't go into much detail. This video already exists. So this is a step-by-step of the entire protocol for this library prep. And Merry Christmas. I went slightly over time, but not too bad. But happy to take any questions, and also happy to show you one of those epitome pipelines if you're interested in it.
Thank you, Dr. Amanda, for that talk. Really very informative. I have a question, though. My question is on during reusing the flow cell. So usually the barcode carryover is really an issue. So when we reuse the flow cell, it's usually recommended to use a different set of barcodes. But does that limitation still apply, given that, for instance, let's say in the first run, we're going to sequence SARS-CoV-2 amplicons, and then for the second run, we're going to reuse the flow cell using the same barcodes, but totally for a different approach, like perhaps using a shotgun metagenomic approach for a sample type where we are not expecting to have SARS-CoV-2 reads. And if that's possible, can we discriminate between the first sequencing run, first reads from the sequencing, from the first sequencing, and from the reads from the second sequencing?
Yeah, that's definitely doable, especially if you're sure there's not going to be SARS-CoV-2 in the second set. It's very simple to just align the data to SARS-CoV-2 and get rid of those reads. But I think you will find that there aren't any. The WASH kit has very recently been upgraded. In the last few months, it's been upgraded. So previously, the WASH kit had quite significant carryover. But now that they've introduced a nuclease, it really does just shred everything that's in there. And if you want to be really sure on that WASH, once you've added the nuclease, you can leave it for a whole hour. I think it only recommends half an hour, if I remember right, maybe 20 minutes. If you leave it for the whole hour, it just decimates everything that's in there. It's very unlikely that you're going to get carryover. And if you do get carryover, it's going to be so low abundance that it's not going to affect any of the pipelines downstream. So I wouldn't worry too much about the barcodes.
Thank you. I have another question. I usually work in the wet lab, so for other applications, like for instance, metagenomic sequencing, can you recommend approaches for host DNA depletion, which is tailored for nanopore sequencing?
There's nothing specifically tailored for nanopore sequencing. So all the kind of standard approaches are all fine. I don't know what is available necessarily over there, but the microbiomes I'm usually doing are rumen microbiomes, and there's so little host in there that we don't even bother. But obviously that's not really, if you're doing things like skin swabs or anything like that, you definitely need to do it. There are some protocols that are based on centrifugation, which are that you don't need to buy a kit, and they're reasonably good. We've used them before on dogs, and I think we started out with maybe 90% dog and got it down to 30. So it did reasonably well. But I can't recommend anything specific because I'm lucky enough to work on samples that don't really need it.
Yeah. Thank you.
I would guess the hello, sorry. Hello. Hello.
Hi.
Yes, Paul.
Hi, Amanda. I'm Paul. I'm the bioinformatician at PGC Mindanao. I think maybe to answer Mitch's concern, I don't know. Maybe the host sequences can be removed by the bioinformatics pipeline, if I'm not mistaken.
Yes, the concern, Paul, is on the distribution of the reads. So it's not efficient if you're gonna sequence and then 90% of your reads is the host. So to save resources. Yeah.
Okay. So it's like you're filtering out the host reads even before it gets onto the sequencing. Is that
Correct.
Yeah, right. Okay.
What size genome is the host? What are you sequencing?
Mammals, like for instance, we're planning to sequence, if we have the resources, bats.
Cool, right? Because there's this new method. There's been a lot of talk of it over the last few days about the real-time selective sequencing. At the moment, I believe it needs to be quite a small reference, but as things get more efficient, you might find that it doesn't. And particularly if you have a GPU to run it on. So this is literally you give the tool a reference, and in real time, as it is sequencing, it base calls so fast that you align while the DNA is still in the pore. And if it doesn't align, or if it does align, you can choose to accept or reject. It will spit the DNA back out and get a new strand if it's not what you want to sequence, which is really cool. And I would love to see that scaled up to kind of a mammalian reference. I think at the moment we're talking a handful of bacteria worth of reference would work with that. But someday, perhaps in the near future, you'll be able to just say don't sequence the bat and it will just spit out any DNA that belongs to the bat, which is really cool. But yeah, for the moment, you probably do need to go with a host depletion method, unfortunately.
Amanda, can I follow up on that? Have you seen the real time, or do you know quite up close people that have used the real time thing? Because I've heard a lot of talk about it, and I've seen obviously the graphs and the talks, and it sounds amazing. I just have never I don't know anyone that's tried it and it worked, so to say.
So I have tried it. I actually went down and visited Matt Luce and did it with them.
Cool.
But I was using it on a microbiome, and it was quite a shared sample because, unfortunately, with microbiomes on Nanopore, you still have to bead them if you want to get the entire population out of the sample. And bead beating has the unfortunate consequence of shredding your DNA. But yeah, we used it, but basically by the time it had aligned the DNA, most of the strand had already gone through. So we saw that there was kind of the distribution of read lengths. You could see that the longer stuff didn't have the pre-patella I was trying to avoid, and the shorter stuff did. But overall, it didn't make a massive enrichment. But I have seen it, not in person, but I've seen the results of it used on a human genome to enrich for cancer-associated loci. And they got a 30-fold increase in coverage over, I think, 100 sites. So it does look like it works. The only thing to note is your throughput's going to be a lot lower because you are spitting out a lot of DNA. And also just the fact that you're reversing the current like that can actually batter the pores, and they tend to die off faster than they would otherwise. So the alternative there would be the Cas9 method. Have you seen there's actually a kit now in the Nanopore store for it, where if you have a region of interest up to 20 kb in size, you can use Cas9 to cut either end, and then the library prep will only work on those cut bits. So basically you wash your DNA, so there's nowhere for the adapter to ligate to except for those cut regions. And then you still end up with a lot of DNA in your flow cell that isn't able to go through the pores, but you don't have this effect of battering your pores. So you still have lower throughput, but it's very specific, and it seems to work quite well. I think there were a couple of posters using it in the Nanopore community meeting, if you wanted to have a look at those.
Cool. Okay, thanks. And I had another question, is when we were looking at what kind of kits to order now for this protocol in the Philippines, I saw that they are planning to discontinue the LSK 109 kit, but then they also seem to say that the new 110 kit is only for single plexing, not for multiplexing, and that kind of baffled me. So I don't know, because I've been using the 109 kit throughout my entire PhD. So if they stop selling that, I'm like, what will I use in the future?
Yeah, I believe they're still selling it, and I don't think there's actually any reason why you couldn't use 110 with barcodes. Okay. I think they're just being careful about what they say before they're sure. But I mean, logically, I can't see anything that's so different that you couldn't use it with the barcode.
That's
I imagine within the next month or two, they will probably clarify that, because
Yeah, that's
if
true.
I remember right, the timeline, they said that 110 is out, but yeah, I think with the wave of things that are coming out in Q1 of next year, they'll probably clarify that.
Yeah, that's true. Makes sense.
I think you'd be fine with the 110, to be honest.
Yeah.
I don't see any reason why not.
No, that's true. And another question, Dev, do you know anyone that's used it? Because I know they also have the field sequencing kits. This is interesting if people want to do it like properly out in the field, of course. But do you know anyone that's been able to use the barcoding kit with the field sequencing kit? I'm guessing not, because you kind of have to use the barcodes, put them on a cold chain. But
yeah, no, I would hope that they're going to bring out a barcoding field kit. I know they don't yet, but to be honest, the rapid kit is fairly field friendly, other than having to be cold. I've used the rapid barcoding kit in a pub before.
Nice.
So yeah, I mean, if you can keep it cold, and if you can access some hot water and some ice, you don't even have to be very specific. I know someone who did it in a hotel room with a cup of coffee for the incubation. So it's not too bad. I've used the field kit once, but it was several months out of date, and it didn't work, surprisingly. But yeah, I would imagine now that the field kit is kind of properly established, that they'll be introducing a barcode version. It just takes,
because for me it just feels so expensive if you can only run one sample. Like
Absolutely.
Nobody's going to do that. So.
Yeah. Like even the quality of DNA that you can get out in the field is not worth a whole flow cell usually. So
yeah.
But I would expect it to be coming.
Yeah, cool, thanks.
Amanda, I have a question. This is Lyre. Can you share to us your experience with ASF here in the Philippines? Like so far, how was the quality of the sequencing?
So yeah, it looks really good. What we did initially while we were actually in the Philippines, because we didn't have a plan, this was October last year, we went to the Philippines, and it was right after the outbreaks started, and we were there to sequence PERS virus. So it's just kind of a, while we're here, this is the bigger problem right now. So in fact, what we did those first few samples was we just extracted DNA from pig's blood and started sequencing it. So we didn't even amplify anything, we just went for it because that was what we had and what we could do on the spot. And it, we ended up sequencing a lot of pig that we didn't need to sequence, but we'd still managed to get whole and pretty accurate looking African swine fever virus genomes out of a couple of those pigs. So since then, while we were there, we did get the cDNA, not the cDNA, sorry, wrong virus. We did extract the DNA and bring it home with us. And we have been working on amplification strategies to make this more efficient for the sequencing part of it. So now we have these 7 kb amplicons across the genome. We've got about 4 amplicons we still need to optimize that aren't quite behaving. And we've adapted the RTIC network pipeline to work with African swine fever. So we expect quite soon to have a bunch of whole genomes from the Philippines that we will be able to release. We're just doing the final tweaks on that protocol. And pandemic permitting, we'd like to come back and get a lot more done actually over there, which would be nice.
Yeah, my next question would be, I always ask this question to the Nanopore people, because so far the people that ask lately don't have the answer. I guess the main issue with the MinION platform is the accuracy or the errors of the reads. But compared to Illumina, of course, Illumina you can get more higher accuracy. So how can we address those issues in terms of sequencing?
So the consensus accuracy is pretty good now. So using the chemistry we were using last year, I'd say the raw read accuracy was 95 or 96%. With the latest chemistry that's available now, I am told that it's up to 98. And the in-house accuracy, they have a new chemistry that hasn't been released yet. They've got up to 99.1%. And that's just on the raw reads, which, if you think about Illumina, that's not even true for them, because they have like their amplified clusters of strands, and they're basically taking the consensus of that. So the accuracy is actually pretty good. And once you've sequenced it, you can bioinformatically get the consensus. And the consensus is pretty good. It's quite difficult for viruses to say exactly how accurate it is, because they tend to be more variable than than is useful when you're trying to find a kind of ground truth. But compared to other available sequences, I've seen it over 99.96, 99.97. So yeah, it's pretty accurate these days. And you'll find that most of the errors that remain after you've polished are in homopolymers, which is where it struggles the most. So it usually guesses the wrong number of A's in a homopolymer, or whatever letters you've got. And what you could do with that is a frame shift correction. So there are tools out there that, if you know what the protein sequence is, it will go through and check that there is no indel causing a frame shift, and it will just correct that by adding the correct number of ends to put it back into frame. And once you've done that, it's there's not much difference, to be honest, between what's already out there and a nanopore assembly. It's getting very good, and once they brought out this new chemistry, it'll be fantastic. I think if they're being completely honest about their 99.1% accuracy, that would be phenomenal. So they're really going full steam ahead on fixing the accuracy problem. So hopefully in the near future that'll be a problem of the past.
Is there an effort right now in the UK to use Nanopore for SARS-CoV-2?
Yeah, there's a huge amount going on at the moment, actually. I'm not involved directly. I've just done some sequencing of the lab strains that one of the groups I'm in are using in their experiments, just to make sure they're not changing over time as they're growing them in the lab. But yeah, there's a lot of people sequencing. I think actually Khalil probably knows the numbers, because he's been keeping an eye on the global numbers. I think the UK might be the highest one. Am I right, Khalil?
Yeah, I think right now you've reached like 100,000 genomes.
Yeah, I think we're world leaders on SARS-CoV-2 sequencing. Yeah.
Cool.
Not to boast, it's not really me doing it.
All right, so we're a bit over time.
Question.
Yes,
Just
go ahead.
one last question. So, for instance, if we're gonna do what you call this, biodiversity profiling, so we can use BAM files, right, to actively call the identity of the, let's say, reads. Can you should we use like whole genome, or are certain sequence files from barcodes sufficient to do the live taxonomic profiling?
So this is actually a good moment for me to bring up my epitome thing. Am I still sharing the screen? I think I am.
Yes.
Yeah.
So this is
before you start, Amanda, have you used epitome?
Yes, this is actually, okay. I said I used the kit in the pub. This is the results from that.
Okay, cool.
Basically, this was at a conference. I was doing a live sequencing demo. So people were having their conference dinner while this was up on the screen, and they were trying to guess what the sample was. So this basically, as your sequencer is running, I believe actually they're going to build this into Minnow very soon, maybe in the next release. But currently what you have to do is, as your sequencer is running, you tell Epitome where the files are going to, and it uploads them in real time and does the analysis remotely. So it tells you sort of basic stats, how many reads you've got for each barcode, your yield per hour, quality scores, and read lengths. But then in this tab, we've got WIMP, which is called What's in My Pot, but we call it WIMP for short. And that in real time classifies your data like by reads and tells you what is in your sample. And it also puts out a tree. This tree isn't very interesting because it's not down to species level. go down to species level and reduce the abundance cutoff. So this comes out in real time. So literally kind of every few minutes as it gets a batch of reads, it'll update this tree and you can watch this tree form in real time. So this is what was happening in the conference, was people were watching this come up and guessing what the original sample was. Please feel free to guess. There is no prize this time, but you can guess if you like. But yeah, you can classify based on reads in real time using this. And Rampart will also do it. So there's kind of a limited database here. This is just a relatively small database that Nanopore put together. But if you have your own database that you're interested in, you could use that as your reference in Rampart, and Rampart will do something similar.
Was it a sourdough starter?
It wasn't, but I really want to sequence my sourdough starter.
Me too. Sorry.
I'm wondering if I could crowdsource that. If you donate, I will sequence your starter with mine.
Exactly.
So if we're gonna make our own database, like for instance, just unique species that's including endemics that's present in our sampling area, would you recommend? Should we include the whole genome, or are specific barcode regions sufficient?
I think I'm an advocate for whole genomes, just because I don't entirely trust 16S and similar things to be as accurate, although it depends what database you've got. If you have the data for all of the genomes, whole genomes, then use the whole genomes. If you've got a more extensive database for a marker gene, you may as well use the marker gene. It's really up to you. You could try it with both, see how it looks and compare them. If you have the data, go for the whole genome. If not,
is it also possible to combine like whole genome sequences for those that we have the whole genome, and just barcode sequences for species that we only have barcodes as for now?
On one flow cell?
I mean, in the database for the
in the database
taxonomic profiling.
I haven't tried it. You could try it. I'm not sure if it would just end up confusing, to be honest. Particularly with your abundance profiles would be confusing. I think I would preferably keep it separate. Again, if you did the sequencing once without doing the analysis in real time, and then try the analysis on both different ones and then decide which one works best, I think that would probably be better than trying to do everything at once.
Another question. In the tree generation, what algorithm are they using? Is this maximum likelihood or?
This is a good question. See, I don't use this for real experiments, just for the pub. So I'm not entirely sure, to be honest. I'm sure that information will be somewhere on the Nanopore community website. You would think it'd say somewhere on here, wouldn't you? But no, it doesn't.
Seems like an NCBI taxonomy tree. I don't know. Maybe it's like an algorithm that NCBI has developed. I don't know. Just guessing from the name.
It is quite nice though. I mean, it's very user friendly. You can click on it and read some information about the species. It's it's a cute little.
I'm interested how they do the classification. Like, what's the
it's using centrifuge
for sequence similarity, or do they have a certain algorithm for?
It's using centrifuge, the classification tool called centrifuge. You can look into how that works. It's basically just a very, very fast classification tool. This is probably not what you want to publish. You probably want to use something a bit more with a more extensive database and a more accurate classifier. This is a little bit gimmicky, but it will work for people who are completely allergic to bioinformatics.
I guess that epitome you can just download off the community website, right?
Yeah.
And this is what we can use for our actual sequencing. Yeah.
Yes, you can. Yeah. So it'll install a very small program, which its only job is to upload the data from your computer to Epitome, and then you go on the website and you can access this, and also you can share it, so you can give this link to anyone else you're working with and they can watch this as well. So yeah, it's quite a nice little tool for collaborations and for real-time analysis.
Lastly, for field implementation, the power requirements for this software, like, is this very, energy, power consuming? Like if we're gonna implement this on field?
It'd be a little bit more than just sequencing on its own, but all it's really doing is uploading. The actual work is going on the internet, so it's not actually happening on your computer. So it shouldn't be too bad. They are also bringing this onto the Mark 1C, which is the sequencer with the screen built in. So if you had the funding to get a Mark 1C, that one is super field friendly. And you can do the analysis at the same time as the sequencing. It has a little GPU in it, so it can do quite a lot.
Thank you.
So,
Thank you so much.
yeah, as you can see with all the questions, I think there's I can speak with for everyone that this has been very interesting, and it's sure helped a lot in preparing us for sequencing for the first time here in Mindanao. So again, thank you so much, Dr. Amanda Warr.
And I better tell you before I go, this was the microbiome from fermenting whiskey.
Yeah.
Nice.
Yeah.
Cool.
All right. Thanks very much for staying around, even though it's so late there. And I best go because I have another meeting in 10 minutes. But it was nice talking to all of you.
Thank you so much.
Thanks,
Bye.
Amanda.
Thanks, Amanda.
Thank you so much.